Hello there, and welcome to this Khan Academy tutorial on AS level biology. In this video, we will look at cell membranes. This is part 1 of the two-part series on this topic. Here are the aims of this tutorial: describe the structure of the cell membrane, define what hydrophobic and hydrophilic means, and understand what is meant by fluid mosaic model, and then describe how small and large molecules enter the cells, and finally look at how temperature affects the cell membranes. So let's get started. You will be familiar with cell membranes from GCSE, but now let's add to the knowledge. Cell membranes are partially permeable membranes, meaning they only let specific substances through. Hence, the membrane acts as a protective barrier. The word plasma membrane is synonymous with cell membrane, and often textbooks refer to this term. Let's look at the structure of the cell membrane. It consists of a phospholipid bilayer. Remember, phospholipids are molecules with a hydrophilic head and a hydrophobic tail. When forming the cell membranes, the hydrophilic heads face outside, while the hydrophobic tails face inside. The word bilayer means two layers, in case you already didn't know that. Hydrophilic refers to polar, water-soluble molecules. So polar essentially means that the molecule has a small charge known as a dipole. Hydrophobic refers to non-polar lipid-soluble molecules. When looking further at the structure of the cell membrane, we see that it consists of integral and extrinsic proteins. Intrinsic proteins span the whole bilayer, while extrinsic proteins are present only on one side of the bilayer. Channel and carrier proteins are examples of integral proteins, while glycoproteins and glycolipids are examples of extrinsic proteins. Glycoproteins are made of protein and a carbohydrate, and they typically act on cell surface receptors. Glycolipids are made of lipid and carbohydrate, and they tend to act as antigens of that cell. Eukaryotic cells contain cholesterol within their bilayer, which provide membrane stability by restricting the membrane moving too much. Do note that cholesterol is not present in cell membranes of prokaryotic cells such as bacteria. Here is an enlarged view of the cell membrane. We described the cell membrane as having a fluid mosaic model. This essentially means that the cell membrane is dynamic. It constantly moves about. The proteins attached to the cell membrane create a mosaic pattern. Now let's look at how common substances pass through the cell membrane. Small lipid-soluble molecules such as oxygen, carbon dioxide, and triglycerides pass through the bilayer by simple diffusion. Monosaccharides, like glucose, enter the cells by facilitated diffusion using carrier proteins. Details about facilitated diffusion will be discussed in another video. Small polar molecules like water can go through the bilayer by simple diffusion, but this is not very efficient. If cells require more water, they use specific channel proteins called aquaporins. Finally, large molecules, including polar and non-polar ones such as hormones, acetylcholine, ACH for short, amino acids, and charged ions enter the cells via facilitated diffusion using channel or carrier proteins, or via active transport, which only uses carrier proteins. Temperature is well known to affect pretty much everything on Earth, and it also affects membrane fluidity. Increasing temperature increases the membrane fluidity, meaning that it moves more dynamically. This is because phospholipids gain more kinetic energy and move more quickly, becoming partially permeable. Note that the membrane fluidity differs to membrane permeability, as indicated by the green curve. If the temperature is too high, the cell's cytoplasm expands due to the water inside, and eventually the bilayer breaks, and the cell permeability increases significantly, and the channel and carrier proteins are also denatured. If the temperature is too low, the cell membrane is rigid. However, the channel and carrier proteins are denatured, making the cell membrane very permeable. And as the graph indicates, the blue line shows that the lower the temperature, the more rigid the cell membrane, and the higher the temperature, the more fluid-like. As the green line indicates that when the temperature is too high or too low, the membrane permeability stays high. Only at the right optimum temperature, which around in our body is 37 degrees Celsius, is where membrane permeability is partially permeable. So here's a recap of the aims that we went through this tutorial. As usual, use them as questions to consolidate your learning. Thank you for watching, and I'll see you in the next one.